Around the world, approximately 463 million people are living with type 1 or 2 diabetes, a disease in which the body is unable to produce enough of the hormone insulin or use it well. Insulin is needed to regulate blood sugar. In 1977, after completing her PhD in cell biology at MIT, Mexican-American scientist Lidia Villa-Komaroff joined a team within the Gilbert Lab at Harvard University attempting to clone the insulin gene. A year later, Lidia became the lead author on a paper titled, "A Bacterial Clone Synthesizing Proinsulin," published in the Proceedings of the National Academy of Sciences, which detailed how to produce insulin from bacteria, instead of horse and pig pancreases, which was the leading method at the time, essentially solving the problem of insulin unavailability. Squared to Lidia, the research was personal. Her grandmother had died from diabetes. Cubed Lidia's contribution in the team was cloning double-stranded DNA that would express insulin. Today, most people who need insulin use a synthetic form made through the methods using recombinant DNA that she and the Gilbert team pioneered. The Food and Drug Administration approved the first rDNA human insulin product in 1982. This form is more biochemically accessible for humans than insulin derived from pigs or cows. Lidia accomplished this groundbreaking work despite discrimination and discouragement she experienced during her higher education journey in the sciences. As an undergraduate at University of Washington, she began as a chemistry major. On the first day of undergraduate chemistry, the professor literally told us, "Look to your right, look to your left. Only one of you will be here by the third term." It was very hostile, she shared in the interview with Shift 7. The early courses were really tough. On one test, the highest score was 10 out of 100 among the whole class. I asked an advisor for help, which was hard for me to do. He looked at me and said, "Well, of course you're having trouble in chemistry. Women don't belong in chemistry." Chemists have gotten their act together since then. She changed her major to biology when the university got an infusion of young dynamic biology faculty, and those classes were fascinating, and they treated us like real people, she explained. John Gordon came in as a speaker. He was a biologist from the UK. I had envisioned all scientists as old men, but he was a red-headed fireball. He won the Nobel Prize many years later. In 1967, Lidia transferred to Goucher College in Maryland, to be with her future husband Anthony Komaroff, who got a job in Washington, D.C. She had been advised to transfer to Johns Hopkins University, but at the time, they didn't allow women in their undergraduate program. Lidia said she enjoyed Goucher College, and highlighted, "The faculty got me a job at NIH National Institutes of Health, in the summers, in a lab run by LORETTA LEIVE, microbiologist." Lidia added, "It was because of LORETTA that I applied to MIT, she encouraged me. I thought I wasn't good enough at math, and that it was all men running around with slide rules. She told me if I wanted to be a molecular biologist, MIT was the premier school for that." Lidia became one of the first few Mexican Americans to obtain a PhD, which she accomplished at MIT. Throughout the course of a very accomplished career, Lidia was part of additional scientific breakthroughs beyond her work on insulin, including demonstrating the first direct evidence that the beta-amyloid protein is toxic to neurons, which may have implications for Alzheimer's disease. She also demonstrated that 3 proteins that regulate gene expression are expressed during the critical period in development where the brain must be exposed to light if an infant is to develop opvision. She was awarded the prestigious Morrison Prize at MIT in 2016, for combining humanistic values with effectiveness in the world of practical affairs, and in particular, in science and technology, and the Women of Distinction Award from American Association of University Women in 2013. She was also selected as the E.E. Just Lecturer for the American Society for Cell Biology in 2000. Throughout her whole undergraduate and graduate experiences, she never met another scientist of color. "The first time I met scientists of color was in 1973, and then I met almost all of them from the country at once," she laughed. Lidia was at a meeting where Hispanic and Native American met and founded SACNAS, the Society for Advancement of Chicanos, Hispanics and Native Americans in Science. There were about 40 PhD scientists attending, all but one were of color. Around 15 of them were Mexican American or Native American. It was then that Lidia began to work on diversity in STEM. She began traveling around the country encouraging more students of color to pursue science, which she does to this day, and she mentored students in her lab. SACNAS has evolved to be a premier mentoring society in the country and the most diverse, she stated. It has grown to also have approximately 10% African American participants. It includes all sciences and cultural sessions as well. She currently serves as chair of the nominating committee for SACNAS. She is also a member of the National Science Foundation's Committee on Equal Opportunities in Science and Engineering, CEOSE. She is now focusing on presenting from a natural scientist's point of view what implicit bias is. Lydia explained during our conversation, perception can result in unintended bias. It's a big data problem. The amount of information that our brain gets, it can't process all of it. It needs mechanisms to ignore some of it. We develop associative illusions based on our culture. The mind makes up shortcuts, and inappropriate associations can result. She continued, implicit biases are habits of thinking that can lead us to misrepresent what a person is based on what we see. In our country, we haven't dealt with our history of racism. Our culture is permeated with senses of what people are worth based on what they look like. We see this with gender too. We all share these biases to a greater and lesser extent. The ability to make shortcuts in drawing conclusions leads to bad decisions. If we are aware of them, we can moderate this effect and improve efforts for inclusion. We're the best species in the world for taking care of our evolutionary deficiencies. Lydia sees her work as far from done. She recently remarked, "We're wasting a lot of talent because kids get the subliminal message that they can't do it, or shouldn't do it." To the power of 4, her message to everyone is, follow your interests. She immediately added, science is fun and interesting. Everything is hard, so why not do something fun and interesting? Plus, scientists are the least unemployed of any segment of the workforce. Here's the fun and interesting description of how Lydia accomplished the cloning of double-stranded DNA that would express insulin. Connect pieces of double-stranded DNA to a plasmid which is capable of replicating inside a bacterial cell. Introduce the DNA plasmids into bacteria under conditions where only one molecule will enter a bacterial cell. Plate the bacteria so that the bacterial cells are separated from each other. Overnight, the bacterial cells will grow into a visible colony of bacterial cells that all are exact copies of the one initial cell. Each resulting cell of bacteria in the colony now has thousands of plasmids inside that include one inserted DNA. Examine the inserted DNA in the cells to find the ones with a copy of the insulin gene. This involves purifying the hybrid plasmid from the colonies and determining the sequence. Add that colony to a gallon of liquid food it loves, and the bacterial cells will duplicate, resulting in billions of them. Then her team had to identify which of those DNA had the insulin gene. Lydia's colleague Stephanie Broome developed a procedure for that. To their surprise, not only did they find the insulin genetic sequence, but they found that the bacteria which contained the DNA with that sequence was making insulin protein. This discovery only happened because a 1978 snowstorm kept them from their lab for a week. Control plates that had been left in the incubator too long colonized when they weren't expected to, showing they were making insulin.